All right, everyone, this is the video for preparing you for the lab this week. In lab this week, you're going to pick a paper. Sorry, this is actually the second slide. You're going to pick a paper that's describing the sequencing of the genome of a bacterial pathogen. And I've given you some questions to sort of help lead you through what they learned, what they did and what they learned in that paper, particularly as it has to do with mobile DNA and horizontal gene transfer, but also just sort of what they learned about the genes that are in the genome. Okay, so this video, the short PowerPoint presentation is an introduction to that. Now there are two short YouTube videos I want you to watch also that have to do with how genome sequencing is done. One of them focuses on Illumina is the company that makes the sort of the main type of DNA sequencer that's used. I want you to watch that. It's complicated, I know. I don't care whether you understand the details of how it's done, but I want you, if you're interested, to get sort of a flavor for how what we call high throughput DNA sequencing is done. The second one is another type of DNA sequencing that we've been using a lot in our lab here at Santa Clara. It's called nanopore sequencing, and it generates much longer stretches of DNA sequence that can help us in assembling sort of the whole picture of a genome sequence. Using nanopore sequencing, it's really quite remarkable. The cost of assembling a complete genome has dropped by probably another order of magnitude. Just to give you a feeling, we published a paper this summer in my lab, Dr. Hess's lab, that talked about the complete genome sequences of 50 E. coli isolates. So just in that one paper, 50 complete E. coli genome sequences. The first E. coli genome sequence that was generated in, I think it was like 1998 or so, probably cost about $50 million to do for one of them. We did 50 of them for roughly $10,000 or so. All right, so these advances in technology have really been pretty remarkable. All right, that's not the main thing I wanted to tell you about. Let's run through genomes, bacterial genomes, and horizontal gene transfer, okay? So at this point, thousands of bacterial isolates and species have had their genome sequences determined using sort of the advanced DNA sequencing methods we have available now. And the important thing to recognize is that most bacterial genomes, not all, but most, are actually quite variable when you compare different isolates of the same species. They are what we call mosaics. There's a conserved region where if you take the 50 E. coli that we looked at, for example, all of them have a subset of the genes in common, right? It might be 1,500 or so of those genes that are very highly conserved, and maybe another 1,000 that are reasonably conserved, right? But then there's a bunch of other pieces of DNA that are highly variable, right? So the main thing that's responsible for this is what we call horizontal gene transfer. So bacteria have mechanisms of transferring DNA from one cell to another, or picking up DNA in the environment. There are then subsequent processes called recombination, as well as mutation and DNA repair. These are part of chapter 9, well, chapter 7 and chapter 9, but we're not going to go into the details on recombination, mutation, etc. But I do want you to know about horizontal gene transfer, okay? So just to provide an example of this, if we looked at 3 isolates of E. coli, these are among the 3 that were sequenced fairly early on, right? The names here are, actually, this particular diagram is just 2 of them. MG1655, which is what we call this classic K12 E. coli, and O157H7, which is a strain that a lineage that causes bloody diarrhea. And you often see, you've heard of this undoubtedly as contaminating spinach, ground beef, fruit juices, things like that. Over the years, there's been a number of outbreaks that have been related to this particular type of E. coli. If you compare those two genomes, if you look on the outside here, red boxes are unique to the O157H7, purple are unique insertions in MG1655, and then the tan boxes are regions where the sequences are, they're basically there, but they're quite distinct in the two isolates. Okay, so the bottom line is the total divergence here, where the sequences are very different, is almost half of the total genome. I'll also point out here that this 4.1 megabases that these two strains have in common is sort of a minimum for E. coli. Most of the strains that we've sequenced actually have over 5 million bases, so they're significantly larger than these, right? So only about a little over half of these two, and this is just looking at two isolates, is highly conserved. All right, so what's this horizontal gene transfer? What do we mean by horizontal, first of all? Vertical inheritance is when one sort of parental cell gives rise to two progeny cells. Each of those gives rise to two progeny cells, right? So that the genome, the DNA that's inherited by the two progeny cells, that's called vertical inheritance. Horizontal transfer is when there's a piece of DNA that comes from a completely different cell, or maybe comes from outside of a cell. It doesn't come directly from a cell. It's free DNA that the cell takes up. So it wasn't there in its parent, right? That's the point. It wasn't there in the parental cell. Okay, so that is horizontal transfer. So in bacteria, there are at least three, and actually I'm going to tell you about a fourth one as well, mechanisms of gene transfer. Conjugation is used by plasmids that encode a specific machinery for getting them from one cell to another. Transduction is mediated by viruses, bacteriophage in this case, since we're talking about bacterial cells, that deliver DNA from their previous host cell into their next host cell. Of course, if this virus ended up killing this cell, we would never know that it delivered that DNA. But if it didn't package its own DNA, if it accidentally packaged a piece of host DNA, then it probably won't kill this cell. Transformation is when this cell takes up a piece of free DNA from the outside, shown here as a sort of lysed cell here, because it had to come from some cell originally, right? But it didn't come directly, it wasn't delivered by an intact cell into this cell. It was free DNA at the time that it was picked up, okay? And the last thing I'm going to tell you about, which you've actually already learned about, is a process by which bacterial viruses, bacteriophage, integrate into the host cell, and they can actually bring functional and useful DNA with them sometimes as well. So that's prophage integration, okay? So conjugation, transduction, transformation, and prophage integration. Okay, so let's quickly talk about each of these, right? And again, the reason I'm telling you about these is these help to shape the structures of genomes, right? Most of that variable DNA that you saw in the previous slide came originally by one of these mechanisms, including that prophage insertion, okay? Now, we're also going to talk about processes of transposition that can happen once DNA gets into a cell, but we'll get to that later. Okay, so transformation is the process of importing free DNA into a bacterial cell. The cells need to be competent. That's just a term that's used to indicate a cell that is capable of taking up free DNA. Now, if that's a natural part of the biology of a bacterium, like it was for Streptococcus pneumoniae, right? The pneumococcal transformation experiments relied on the fact that that species is naturally capable of transformation. But it's also possible to induce this in the laboratory by either types of chemical treatment or a process of electrical shock called electroporation. Okay, so this is all natural transformation, right? So in order for that to happen, I'm not going to go over this in great detail, there has to be a machinery in the envelope of the cell that will bind to the DNA and then give it a pathway across the cell envelope and into the cytoplasm. Okay, so this machinery is the competence machinery, right? And this is present in Streptococcus pneumoniae, for example, right? So it takes up the DNA. And then if that DNA, remember, this is just random DNA that's outside. Typically, in order to be maintained in the host cell, there has to be a recombination event that actually connects this DNA with the host chromosome, right? And it can, usually it happens by a process of recombination called homologous recombination, where there are sequences on the incoming DNA that are very similar to the sequences on the chromosome. And then there's a machinery in the cell that actually allows that recombination to happen, okay? And then often a lot of that incoming DNA or the DNA that's then replaced after recombination is degraded. This linear DNA is not going to be maintained in the cell. It's going to be broken down, okay? So that's the basic process of transformation. Now, if that DNA happened to be a plasmid that had its own origin or replication, et cetera, then it might be able to be maintained separately, right? But if it's a linear piece of DNA, it probably won't be. When we do this in the laboratory, we do it with intact plasmids, and our goal is actually just to get that plasmid into the cytoplasm where it can then take care of replication itself. All right. Conjugation is the second mechanism we talked about. Conjugation is when a plasmid intentionally sets up a machinery to go from one cell to another, okay? There's a link here you can click on that will tell you more about the details of that. This is a famous colorized image showing the, remember when we talked about pili in bacterial cells and we said there's such a thing as a conjugal pilus or a sex pilus that will reach out, attach to another cell and then bring them back in contact with each other so that the delivery can happen. This is an example of that, right? So this plasmid has this this cell has this F plasmid. It's a conjugal plasmid. It encodes the production of these sex pili. This comes in contact with another cell, pulls them together, and then there's a bridge that occurs between these, a channel that's generated between the two cell envelopes to allow this plasmid to go into the new cytoplasm. This particular plasmid uses this process of rolling circle replication to essentially spin off a new copy that's going to then reside in this new cytoplasm, okay? And then, of course, it will be able to make its own conjugal pili because it will have the genes for that conjugal pili are encoded on this F plasmid. The paper that I was telling you about that we published this summer with the 50 E. coli genomes was showing that antibiotic resistance genes in E. coli are almost always carried on F plasmids, right? So being able to move from one cell to another is a key process for spreading antibiotic resistance in populations of bacteria. All right, conjugal pili can be really important in the biology of pathogens. They can have other, not just antibiotic resistance genes, they can have other genes that function in pathogenesis as well. Just to give you an example of it, there's a bacteria called Bacillus cereus, which is a very common soil bacteria, spore-forming bacteria. It's in this genus Bacillus, etc. It can cause food poisoning, but it's found pretty much all over the place in soil. Has no plasmids in it, right? But if you put the right plasmids in it, namely these two plasmids, these two virulence plasmids, you turn this non-pathogenic Bacillus cereus into Bacillus anthracis, which causes the disease anthrax. And these plasmids encode the genes for a capsule to protect them from the immune system. They encode the genes for anthrax toxin here, which is a very potent toxin for attacking host. And that host could be humans, it could be livestock. We'll talk about, you may see this referred to in some of your papers you're going to work on. This PAI stands for pathogenesis associated island. That's a region of DNA in this plasmid and another one in this plasmid that contains a lot of genes involved in the virulence of this pathogen, Bacillus anthracis. And all the key ones in Bacillus anthracis for causing anthrax specifically are located on these two plasmids. So you go from a relatively harmless bacteria, not completely harmless, but mostly, to a very dangerous pathogen with these. Now, if you're an insect, this is probably even worse for you because there's another pathogen, an insect pathogen, that's also created from Bacillus cereus by virtue of another plasmid. This is the plasmid that encodes what's also known as the Bt crystalline toxin, right? These are the cry genes on here, allow for the synthesis of a toxin that is very lethal to the guts of insects that swallow the spores of this Bacillus thuringiensis. So when Bacillus thuringiensis makes spores, it attaches this toxin to the outside of them. So this is a bacteria that's often found in soil and in plants. And when a bacteria eats a leaf of a plant that has a bunch of these spores on it, these the spores will germinate in the gut at the same time as this crystalline toxin is attacking the gut of the insect, which allows this bacteria then to proliferate and kill the insect and generate a new round of spores. So again, all the harmful genes are encoded on this plasmid. These are all conjugal plasmids. All right, hang with me here for a few more minutes. Transposons are genetic elements that can be present on plasmids. They can also be present on chromosomes. We're including them here with this discussion of mobile elements because they usually encode, or they often can carry antibiotic resistance genes. So when we see, this is spelled wrong, this should be lactamase. When we see these genes on plasmids, it's because these transposable elements carried them there. Okay, but they can also carry them onto chromosomes. I'm not going to go into this in great detail other than to say that these are mobile genes. When they were first discovered, they were called jumping genes. They can move around in a genome and jump onto a plasmid, jump off of a plasmid. Plasmids can carry them into new cells, etc. So they're important mobile elements. The third mechanism of mobility of horizontal gene transfer is transduction. And transduction, what we have is a bacteriophage that when it infects a cell, it's really sloppy in picking up the DNA for its new capsids. So sometimes it accidentally packages the host DNA into it instead of viral DNA. And so you have viral particles that go out, and they infect a new cell, they get injected into this new cell, but there's no viral DNA there. So it's a non-productive infection, but this piece of DNA can then recombine with the host chromosome to then diversify that host chromosome. Okay, so essentially pieces of this previous host chromosome are shot into this new cell where they can then recombine. The last one I told you about was prophage, so viral genomes that integrate into host genomes. Remember we talked about lysogenic phage. in which the virus integrated into the chromosome as a natural part of this life cycle. So it can stay there and replicate along with the chromosome. What happens frequently is that you get mutations in this. And now that what was formerly a viral genome is now locked into the chromosome and it becomes part of the chromosome. So you see remnants of viral genes there, but sometimes that viral genome is carrying pieces of other DNA from recombination, for example. So that is actually remarkably common. I don't think we've ever sequenced a bacterial genome, certainly not an E. coli genome, that didn't have multiple prophage inserted into it. All right. It is relevant to pathogenesis because prophage have been discovered that are actually carrying virulence-related genes on them, including in Shigella strains, the Shiga toxin. I'm sorry, not in Shigella strains. Shiga toxin is found in Shigella strains, but it's also found in some E. coli strains. It's called Shiga toxin just because of the guy who discovered it. It's important in the process of attacking the host, and it is actually carried from one strain to another on a phage. Cholera toxin is also delivered this way. So that can be really important. So when you're looking at your papers on bacterial genomes, I want you to be on the lookout for those various types of horizontal gene transfer mobile DNA that's contributing, especially if it's contributing to the pathogenesis of that particular bacterium. Okay, that is all for this video. Thanks.